Hi, this is Sadashiva Pai from Science Mission. So today we have Dr. Biplab Dasgupta with us. Dr. Dasgupta is an Associate Professor of Pediatrics and a member of a Division of Oncology at the Cincinnati Children's Hospital. So his research focuses on understanding mechanisms of cancer development and progression. So Dr. Dasgupta has published several articles in peer-reviewed journals, and he has several grants to his credit. He is also in the editorial board of few of the journals. Welcome, Dr. Dasgupta, to Science Hangout.
Hi, thank you for the invitation.
Thank you. So can you please talk about yourself, your background and all?
Where do you want me to start from?
Wherever, like your education, everything, maybe in few sentences.
So I grew up in a small town in India, and although I went to a very good school in India, it's close to Calcutta, which is called Kolkata. And then I finished my college and my master's in India, and then I did my PhD from Indian Institute of Chemical Biology, which is a flagship research institute of Government of India. So after finishing my PhD, I moved to the US to do my postdoctoral training at Washington University in St. Louis. My primary interest during my postdoctoral training was neuroscience. So basically brain development and the development of brain tumors. So I studied, cancer cell signaling, also signaling in normal neural cells, the glial cells, called astrocytes. And I also studied, for example, the role of different tumor suppressors. The first tumor suppressor that I worked on was called NF1. So I studied the role of NF1 in the development of optic pathway gliomas in mice. So this is since then I also worked on the role of this bioenergetic sensor. It's called AMP kinase. This is a central energy sensor of our cells. And this is a very interesting enzyme, a very controversial one though, because a lot of its function was not known when I started working on it, particularly its role in the development in cancer. I started working on this enzyme, and we made several important discoveries. We found, I'm going to tell you in my slide, in my presentation, what the discoveries we made on the role of APK in cancer, but we also found that APK is highly expressed in the brain and many other tissues. And it inhibits mTORC1, and many biosynthetic processes. We figured out the role of APK during brain development. And then after finishing my postdoctoral training, I moved to Cincinnati Children's Hospital, which is, I would say, the top 3 pediatric hospitals in the country. So here I moved in 2009 as a tenure track assistant professor. And then I rose to the ranks, and now I am a tenured associate professor at Cincinnati Children's. And my research is primarily is focused on understanding the role of AMP kinase and the bioenergetics and the metabolomics of human cancer. And we use primarily brain tumor as a model.
Okay. And can you please talk about your lab and your students and all?
So I run a really small to medium-sized lab. My lab has, generally we have about 2 to 3 postdocs. We have about 1 or 2 graduate students and about 1 or 2 research assistants. And I always had the fortune of, undergraduate students who, for whatever reasons, they kind of like, I don't know, like me or not, but at least like the kind of research we do. So I always had a bunch of undergraduates who would approach me and they were training my lab. I think I had probably trained between undergraduates and some high school students also. I would say about 30, 35 of the students who trained various times in this 9 years in my lab.
Okay, that's good. And I know you are busy with the research, so can you please talk about your extracurricular activities, like your games or you play or whatever, like just to take your mind off of the hard work?
Yeah, so it's not easy to take work away of our minds. I mean, we leave our offices, but at the same time, the work never leaves us, right?
Yeah.
I mean,
Yeah.
this is just a nature of the beast. I love to play tennis. I honestly don't get much time, though. But I am a religious runner. I try to run as much as I can, both outdoor and sometimes if the weather is too harsh, I run indoor at the gym. I do some freestyle, little lightweight workouts, which I try not to miss. I would say 4 to 5 days a week I do this. But I try to run mostly in the summer or throughout the year, and I take my running shoes when I go to meetings and stuff. Besides this, I had a hobby. I'm trying to revive the hobby, which was photography. I have a very expensive camera. And, so I have the gadgets. I just don't have the time. The iPhone is replacing all cameras, as you know. So I have still plans to revive my hobby as a photographer. I really enjoy taking, doing photography.
Okay, and coming back to the research and your career, so can you please talk about some of the people who supported you, mentored you, helped you in your career?
So I would like to start with my, a little earlier. I had the fortune of being tutored by some of this fascinating teachers in India. Actually, there was one teacher in my high school, and there was at least 1 or 2 teachers in my college, and at least I would say 2 teachers during my master's who taught us, evolution, biochemistry, genetics, molecular biology. So those, I would say some of these teachers really inspired me, not only to learn how to teach, but to understand how exciting the subjects are. I would say they played a very important role in pushing me in coming to basic science. I mean, when people ask me what would you become if you didn't become a scientist, I say I would probably become a neurosurgeon. That was, I thought I was going to be that, but anyway, that didn't happen. So I came to basic science and I fell in love with it. And if you ask me today, that those teachers made really tremendous contribution in to what I've become today. I would say during my postdoctoral, during my PhD time, I think my PhD mentor, Dr. Roy in Calcutta, he was very inspirational. He was very hands-off, and he gave the opportunity. He gave me the freedom to do anything, whatever I wanted to do. So I learned a lot. I failed a lot, but I learned a lot in this lab. I would say I got excellent postdoctoral mentors in WashU, Dr. Gutman, Dr. Milbrandt. So both David and Jeff are excellent mentors. They're different styles, but I learned a lot from both labs, and I was able to publish good work from both of the labs. And then I would say when I moved to Cincinnati Children's as independent faculty, it's just a huge deal to start a new lab and to get your first grant funded. I will name one person his name is Kenneth Campbell. He was really instrumental in guiding me and mentoring me to get my first R1. I'm not sure what where will I stand without Kenny's really active participation and help. And Kenny's not in our department, but Cincinnati Children's is a place where people help out each other. It goes, it cuts through the boundaries of the department actually. Kenny really helped me. So I would say, his mentorship is something that I will never forget.
Okay. And coming back to the work and research, so can you please talk a little bit, give some preview of what you are talking about, and give your presentation, please?
Yeah, so there are many projects in the lab. In the short time, I've chosen to discuss about just touch upon 2 or 3 projects which are related. These are all brain tumor related projects. I have other projects, both cancer related and normal brain development projects. For the benefit of time, I will not be discussing that today. But today, what I'm going to tell you is, when we started our lab, there was a big question about this specific molecule called AMP kinase, whether this molecule, this is a bioenergetic sensor, whether the sensor is a tumor suppressor, or let me rephrase it, whether it suppresses cancer or tumor development, or it plays a pro-tumor role. So there was a lot of controversy on the subject, and that is the subject that I found very exciting and interesting and challenging. So we took up the matter, and we started doing experiments. And I'm going to show you what the field was like 10 years ago and how the field has evolved because of research from our lab and many other labs that worked during this 10 years to show that the field is kind of evolving very fast. So I can show you when I get started.
Okay, sure. Go ahead.
So do you want me to go to the slideshow?
Yeah.
Okay, cool. Let me know if you can see it. Can you see it?
Not yet, no.
Okay, wait. Let me just...
Yeah.
You can see it right now?
Yeah. You can go to the presentation mode. Yeah.
All right, cool. So what you're seeing here now are on the left and the right are, you can see the presentation, right?
Yeah.
Okay. So on the left and right, you can see there's the left. So we have cleared the brain. This is called a brain clearance technique. So we cleared the brain and it made it kind of transparent. In the left, you can see the normal blood vessels in the brain tissue. You can see there's a certain structure and a certain form and a certain pattern, right? On the right, you can see that the white circles are the tumor cells, the brain tumor cells. These are human brain tumor cells that we implanted. And you can see in the green are this really highly abnormal tumor vasculature. Okay, this tumor vasculature is abnormal. It is already known, so we are just to the brain clearance. We can actually see this very clearly in 3D. That how strange and abnormal this vasculature inside the tumor is. And the red dots are potentially hemorrhage because brain tumors, the blood vessels are very abnormal and they rupture and they release blood, and it's called hemorrhage. And hemorrhage is a very common feature of glioblastoma, the brain tumor that we study. So here we are showing that the vasculature is highly abnormal inside the tumor, and there is hemorrhage inside the tumor, right? So in the next slide, what I'm going to show you is what you're seeing is a cartoon of a cancer cell and the cancer cells. Can I use my pointer? Or
Yeah.
so you can see the cartoon of a cancer cell. You can see these are some of the genes that are amplified, the potentially oncogenes, and these are the tumor suppressors that get deleted in cancer. So any cell, particularly the cancer cells, the needs are higher. They have two essential needs. One is a biosynthetic need, and one is a bioenergetic need. So they need to produce a lot of energy and use that energy to synthesize a lot of macromolecules. And in doing so, in order to do all this, cancer cells undergo metabolic reprogramming, and there's a rewiring of the signal transduction pathways and several metabolic pathways. And there's a crosstalk, a constant crosstalk between the metabolic pathways and the signal transduction pathways. And I think it's a cancer cell survival really critically is dependent upon a balance. Because the immune system is really hostile to the tumor cells, so they have to evade from the host immune system. Okay, and so the goal of the body is to kill the cancer cell, and the objective of the cancer cell is to proliferate and survive. Okay, so as they proliferate, the tumor actually becomes very big, and there's a lot of stress that builds inside the tumor, and there's a lot of natural cell death inside the tumor, right? So the tumor has to make a balance because it has to live, and if it wants to live, it wants to spread. So I think this appropriate balance between proliferation and survival really determines if a tumor is going to survive or not, or in other words, if it will be able to spread the disease and kill the patient. So so I want to tell you about this one kinase that is really important to provide and support the bioenergetic need of the cancer cells. And this enzyme is called AMP activated protein kinase, or AMPK. This actually activates the bioenergetic pathways while it actually blocks biosynthetic pathways. So the way AMP kinase works is it's kind of like if you imagine your cell as a car, and it has got a gas pedal and it's got a brake. So the gas pedal is, for example, the molecules that drive the car, and these are some of the molecules that drives biosynthesis. For example, mTORC1 is one of these gas pedals. But if you keep your foot on the gas pedal for a very long time, soon you will run out of gas. So there has to be somebody, to apply the brakes. AMP kinase is the brake. So it allows the cells to constantly gauge how much of fuel is there. And depending on how much fuel is there, it actually balances the speed of the car. So if you're a cancer cell, it allows the cells to survive during periods of energy stress. Okay, so it slows down the cell, and when there is plenty of energy or food available, the foot is taken off the gas pedal. So AMP kinase, as an analogy, AMP kinase will be inactivated, and the cells can do all the biosynthetic processes. So AMPK is present in all organisms. It is an evolutionary conserved cellular energy sensor, and it's a hub for cellular metabolic control. It's a heterotrimer consisting of alpha, beta, and gamma spirits. There are multiple alpha, beta, and gamma spirits. So when in a normal cell, which maintains a high ATP to AMP ratio, it causes an allosteric inhibition of AMPK. But when there's energy stress, there is allosteric activation. AMP binds to this gamma subunit, and there is allosteric activation of AMPK. What it does, it allows a conformational change in this catalytic alpha subunit that allows phosphorylation by an upstream kinase called LKB1. And this actually causes full activation of AMPK. There's another method of activation of AMPK. It's potentially energy independent. We are not completely sure about it. It is calcium dependent, and it is done by a kinase called CAM kinase beta. This calcium calmodulin kinase beta. And it's also, there are a couple papers that showing that an enzyme called TGF beta activated kinase TAC1 can also phosphorate AMPK alpha at the same site to fully activate it. So when AMP gets fully activated, there is energy homeostasis. So what happens is that there's AMPK phosphorylates a lot of substrates, and it causes both immediately and chronically. Transcription is also affected. So ultimately, there is energy homeostasis. So this is how AMPK protects cells during energy stress. So as I said before, that AMPK inhibits biosynthetic processes or anabolism. Anabolism consumes ATP, so when there is energy stress, AMPK will block anabolism so that not a whole lot of ATP is consumed. And how it does, for example, it inhibits fatty acid biosynthesis. It inhibits cholesterol biosynthesis. It inhibits ribosome biosynthesis, protein synthesis by phosphorylating all these substrates and inhibiting them. At the same time, AMPK also promotes catabolism. Catabolism of the process is where more ATP is produced. So AMPK, on one hand, suppresses ATP consumption, and the other hand, it actually allows it production. And the way it does it is actually augments fatty acid oxidation to produce ATP. It actually promotes glycolysis, although how it does promote glycolysis is a bit controversial. It promotes energy production by augmenting mitochondrial oxidative phosphorylation, although how it does is again very controversial. It also allows the cells to survive stress through a mechanism called autophagy. And it also cells by various other mechanisms. It allows cells survive during hypoxia and nutrient stress. Some studies say that AMPK also helps in cell migration and resistance to maybe drugs and xenobiotics. So because AMPK blocks these biosynthetic processes, for example, lipid synthesis and protein synthesis by inhibiting mTOR, for a very long time, as I was stating before, for a very long time, agents that activate AMPK, for example, metformin, particularly metformin, is a drug that is used for the management of type 2 diabetes. I think it's probably the most well-known drug in the field of diabetes. It's prescribed in millions of people for management of type 2 diabetes. And people have used metformin, other agents like ICAR, and many other agents. So all these agents actually, if you throw these agents to the cells in a cell culture, they will activate AMPK. And so what these agents did, it one hand activated AMPK, and it also suppressed proliferation. And it was viewed for a long time that this suppression of cell proliferation by these agents is because of AMPK activation. So that's why AMPK was thought to be part of the tumor suppressor pathway. But whether these growth suppressive effects of these reagents were actually going through AMPK or the suppressive effects really specific for AMPK or not, it's not really thoroughly studied. So for many, many years, I would say, there are several papers that showed that that AMPK is part of our tumor suppressive pathway. So when we started working, we found that cancer cells actually express a lot of many cancers express a copious amount of active AMPK. So we questioned whether this compounds, this ICAR, metformin, and various other compounds that actually suppress cell proliferation, is the effect of proliferation AMP dependent or independent? So as you can see here, this is an untreated cells, and these two are this is a control cell, And this is a cell that doesn't express AMPK. We actually silence the AMPK here. And so the cells with this, with this brick pattern are the cells that we have silenced the AMPK. But you can see that when you add these reagents, this ICAR and metformin, you can see that regardless of AMPK's presence or absence, these reagents are capable of reducing the proliferation from control to here and from control to here. So it doesn't matter. This compound does not discriminate between cells that have or do not have AMPK. So it clearly shows that the agents, the agents do suppress cell proliferation, but it does it, independent of AMPK. We have done the same experiment in normal cells. So these are brain tumor cells, these are normal cells, and we found the same effect, that the suppressive growth, proliferation, suppressive effect of these reagents are independent of AMPK. Now, we took the study in vivo, and you can see here, so these are tumors that are grown in the flank of animals, of mice. You can see it's a controlled tumor. Now, if you treat these animals with ICAR or metformin, the same agents that activate AMPK, you can see all these 4 tumors are smaller than the control, okay? But the important point here is that the cells, the tumors that were derived from cells that we have actually silenced the AMPK, they're also suppressed. Their growth is also suppressed by these reagents, clearly showing that AMPK is probably not required for the growth suppressive effect of these cancer cells, growth suppressive effect of these reagents. So we also found that metformin has been shown to really block mTORC1 through AMP kinase, because this was the pathway that was known before, that metformin will activate AMPK, and then through this TSC2 and Raptor, metformin will actually inhibit mTOR as as part of metformin's growth suppressive mechanism. But what we found is that here, the 2 readouts of mTOR C1 activation is phosphorylation of S6 and phosphorylation of 4-EBP1. And you can see that this is a control NT cell, and this is an AMPK silent cell. You can see that metformin can block phosphorylation of S6 regardless of AMPK, right? So again, showing mechanistically that the effect of metformin in blocking mTOR can be independent of AMPK, at least in this context. So, so metformin is known as a mitochondrial complex 1 inhibitor, and there is, there is no ambiguity about it. So as you can see that when you actually treat the cells with metformin, OCAR is the respiration of the, as a readout of the respiration of the cells. So you can see when you add metformin, the respiration goes down here. And as the respiration goes down, the cells try to compensate for the loss of ATP in respiration by actually boosting glycolysis. And ECAR, as you can see, is a measure of glycolysis. So as you add metformin, the respiration goes down while the glycolysis goes up. So even this effect of metformin, this compensatory glycolysis, is also, we found, is independent of AMP kinase. You can see that here, metformin is, adding metformin causes this compensatory increase in glycolysis, and it happens both in AMPK proficient and AMPK deficient cells, clearly showing that all these effects of metformin are AMPK dependent. So we have published this paper in 2014 in PNS, and we want to move on to see what's the status of AMP kinase in the tumor that we are really interested in. It's called glioblastoma. As you know, it's a highly lethal disease in both in the adult and the pediatric population. Nobody really survives this disease. So what we found, so we analyzed the tumor cell atlas. So this is where you can see the expression. Actually, you can examine the expression level of the genes of your interest. So this is GBM glioblastoma. This is normal brain. As you can see that, so these are the different subunits of AMPK, alpha-1, beta-1, and gamma-1. So you can see that compared to normal brain, in the tumor, AMPK expression is pretty high. Now, we found the same thing in normal human astrocytes that you can actually buy, and we actually maintain them in our lab. Compared to normal human astrocytes, so these are the brain glial cells, but the normal counterparts. Compared to these cells, you can see that the patient-derived primary GBM lines, they have a high amount of active AMPK. And AMPK actually phosphorylates a couple of substrates that are shown here. One is acetyl-alkyl carboxylase and Raptor. So more phospho-AMPK means more active AMPK, and more active AMPK means more phosphorylation of the substrates, as you can see here. We saw the same thing when the protein lysates of fresh GBM tissue that we collaborate with neurosurgeons across the street and also neurosurgeons and other labs from throughout the world. And we procure those tissues, frozen tissues, and we make protein lysates, and you can see that in most tissues compared to the normal brain, these first two are the normal brain, this AMPK pathway is pretty active. These samples, G3 and G8, these samples are degraded, so you cannot see proteins here. So this is an immunohistochemistry of human glioblastoma. You can see this brown staining showing high AMPK activity in the tumors. This is a quantification of the phospho-AMPK or active AMPK signal compared to the normal brain. This is tumor, this is normal brain, so it's showing very high AMPK signal. And this is a genetically engineered mouse model of high-grade glioma. And you can see in some of these tumors, AMPK activity is pretty high. So you can see anywhere you have a high-grade glioma, be it in a mouse brain or in a human brain, you can see a high level of AMPK activity. So we want to know, what is this high AMPK activity doing in this brain tumor cells? So as you see here, so this is a primary brain tumor cell that is temporarily, we adhered them to monitor apoptosis and mitosis. So this is a normal cell. So we can see that if you can keep an eye on the cells, particularly the one with this green dot, you will see that cells will just pop up and will divide into two cells. Just a cancer cell normally divides. So you can see it just divided into two cells. Now, if you see this video, so these are the cells that we have silenced AMP kinase. So you can just keep an eye on anywhere, but particularly I would like draw your attention on this blue dot, on this yellow dot circle, see what happens. What you will see, that these cells not having AMPK will just pop up and burst and showing a sign of apoptosis. So loss of AMPK causes apoptosis of this brain tumor cells. So we have done this experiment in many GBM lines. So you can see that this is non-target, the control. And if you silence AMPK, these cells really don't want to grow very well. And we have used AMPK. This is shRNA through lentivirus. This is siRNA. This is more acutely downregulating AMPK. We saw a very similar effect. We used a dominant negative AMPK and also CRISPR. So by using multiple methods, we show that in glioblastoma cell lines, primary cell lines that we derive from patients, AMPK is really required for bioviability of the cells. So we took this in vivo, and as you can see that, so these are the control, this is the control animals, and you can see that they were transplanted with luciferase labeled cells, so you can monitor the growth of the tumor longitudinally. You can see this is day 6, there's a small tumor developing. By day 9, almost everybody got tumor. By day 12, you can see almost all animals getting tumor, but if the cells don't have AMPK, the tumor growth or development is significantly delayed. As you can see here, the control shRNA by day 17, everybody has got a huge tumor and they have to be all euthanized, while the tumor growth is delayed by many days. You can see this is about 35 days they got a tumor, and these two animals essentially remain tumor-free. So this is the survival curve we can showing that if you, the 2 lines, we have done it on 4 lines now, that the control mice with controlled tumors with AMPK, that they die much sooner than mice with tumors where we have suppressed AMPK. So this clearly shows both in vitro and in vivo, AMPK plays an important role in tumor growth and survival. So what is the mechanism? We did RNA-seq analysis to understand what is going on in these cells in terms of expression of genes when we actually knock down AMPK. And to our pleasant surprise, we found that one of the pathways that was downregulated, so this is AMPK proficient, this is AMPK deficient, there's two different lines. You can see that the glycolysis pathway is downregulated when you actually silence AMPK. And there are several genes that we found to be downregulated when you suppress AMPK. And we have validated these results down both the RNA and protein levels, both in vitro and in the tumor. This is the pathway analysis to show that central to this pathway is HIF1 alpha, that is critical for the transcription of almost all genes that are required for glycolysis. Another pathway that we found downregulated following AMPK silencing was the mitochondrial pathway. As you can see that the mitochondrial RNA metabolic process, mitochondrial transcription, mitochondrial beta oxidation, these were all downregulated. It was kind of known that AMPK regulates mitochondrial function, but it was still very controversial about how AMPK, actually mechanistically, how AMPK regulates mitochondrial function. So we found that the several genes that are downregulated. One of them was critical, called TFAM, which is found to be, this is a master transcription factor for both mitochondrial DNA replication and mitochondrial biogenesis and mitochondrial transcription. So in the pathway analysis, we found that there is a transcription factor called GABPA, GA binding protein A, and also called NRF2. This transcribes this transcription factor TFAM. So we found that this is the pathway that is probably downregulated in AMPK silent GBM cells. And all these results that we found, all the genes we actually validated both at the protein and RNA levels. So we did some functional assay. So ECAR, again, is a measure of glycolysis. OCAR is a measure of respiration. So as you see, we use two different silencing RNAs against AMPK. You can see if you knock down AMPK, glycolysis will go down, mitochondrial respiration will go down, oxygen consumption will go down. And so when these two bioenergetic processes become slow or goes down, of course, the energy production will go down. And so through mass spec analysis, what we're showing here is that the ATP-AMP ratio goes down, the ATP-ADP ratio goes down, the NADH and the NAD, this ratio also goes down. Because these ATP, ADP, and NADH are produced from these pathways. Now, cellular bioenergetics it may or may not be similar to tumor bioenergetics, so we want to make sure that what we found in vitro is also true in vivo. So as you can see, we grew tumors that are proficient in AMPK or deficient in AMPK. We took this tumor tissue out and repeated the same experiment, and we basically validated our in vitro data in vivo, which we can call it ex vivo, because we took the tumor tissue out and measured the glycolysis and oxygen consumption, validating that AMPK is required for tumor bioenergetics in the tumor. So what we have shown so far that AMPK probably augments HIF1 alpha and GABPA transcription, and this probably regulates glycolysis and mitochondrial function. We have done, HIF1 alpha and GABPA function was confirmed by promoter activity assay, by CHIP assay, silencing RNA overexpression and rescue experiments. So essentially what we found that AMPK somehow positively drives these two transcription factors, HIF1 alpha and GABPA, and each of these are master regulators of glycolysis and mitochondrial function to ultimately regulate glioblastoma bioenergetics and survival. So we couldn't find any direct connection between AMPK and HIF1 alpha and GABPA, so we thought this potentially common player that is functioning between AMPK and these two molecules. So so this is let me just step back a bit. So this is one experiment that we did to show that this is a single molecule fish, a small molecule fish, showing that the HIF1 alpha transcripts. So each red dot is potentially a single HIF1 alpha mRNA. As you can see, if the control cells that have AMPK, there is a lot of HIF1 alpha transcript, but when you knock down AMPK, the HIF1 alpha transcription goes down. So coming back to what is this molecule that is linking AMPK and interferon alpha? Through several analysis, including informatic analysis and chromatin IP experiments, and ChIP-seq data, and looking at the ChIP-seq data that is already published by other labs, we found that this is potentially another transcription factor called CREB1. Now, CREB1 is actually very interesting because CREB1 is actually expressed very highly in GBM compared to brain. But there are other publications that were that came out long before our studies that shows that in GBM, CREB phosphorylation is very high. We actually found a site in CREB is the serine 133 and this sequence looked a potential target for AMPK phosphorylation because it really matches the AMPK consensus site. So we hypothesized that AMPK potentially phosphorylates this serine 133 in CREB and activates it, and that phosphorylation causes CREB activation. So bottom line is essentially that's what we found. So this is controlled tumor. This is AMPK silenced tumor. You can see this phospho-CREB signaling goes down in the tumor. We do a reverse experiment. We take the normal human astrocytes and we do AMPK activation, and you can see the phospho-CREB levels goes up. So AMPK activation causes increased CREB phosphorylation, and AMPK silencing got decreased CREB phosphorylation. So when we actually use now, we ask a question now: if CREB is important, then if you now knock down CREB, will the glycolysis and respiration go down? And exactly that's what happens. If you knock down CREB, the glycolysis goes down and the respiration goes down in these cancer cells. And we actually next took one more step to understand if this phosphorylation event is really critical. For example, if you can mutate this site and such that AMPK cannot phosphorylate serine at 133, then you'll probably be able to rescue the animals. For example, you will actually block the tumors to grow, and the animals will live for a longer time. So that's exactly what we found. So if you actually do a site-directed mutagenesis of this site from S to A, now this site cannot be phosphorylated by AMPK. And you can see that while the control animals die by day 17, many of these animals where we transplanted tumors expressing this mutant CREB now lives longer. So this was the pathway that we finally proposed, that AMPK phosphorylates AMPK, although it inhibits mTORC1, at the same time it phosphorylates CREB and regulates GBM bioenergetics and survival. We recently published this in Nature Cell Biology last year, and my new postdoc is actually is continuing this project to work on the next step of this project. So what we have shown so far, this is what I think about how AMPK functions in a tumor. I think it's a context-dependent tumor suppressor. When I say tumor suppressor, it never gets deleted. I'm saying it's got a tumor suppressive function, which is context-dependent. So what I think is that during the first days of cancer formation, the tumor formation, I think the cells are proliferating really fast, and during that time, maybe there is a low AMPK activity in some of the cells. There could be some AMPK pools, active AMPK pools, that are regulating cell cycle surveillance and proper mitosis of the cells. But as the tumor grows, and with the rise of cellular tumor heterogeneity, and particularly during advanced tumors, a lot of stress builds up. Okay, so stress builds up in the tumor, and it requires a balanced proliferation and survival. And particularly when the tumors become really stressed out and big, and there's a tremendous amount of nutrient and oxygen flux and going on. There's hypoxia and tumor acidification and oxidative stress. I think during this time, AMPK activation in potentially, not maybe all regions of the tumor, at least in subdomains of the tumor, it provides metabolic plasticity to the tumor and gives a growth and survival advantage to the tumor. So we published this study, and as I said, we are continuing with some unanswered questions of the study now. I'm going to switch quickly to another project that we've been working on. We just actually published a study in 2000, I think a couple of years ago. So the cells you're seeing here are, considered as the cell of origin of brain tumors, or astrocytoma or glioma. So these cells are astrocytes and neural stem cells, or this invariantly differentiated glial cells. It could be oligodendroglial cells. What you're seeing here is the human ventricular system. Is in the adult brain. You can see that this is a lateral ventricle, and this is the fourth ventricle. And this lateral ventricle, you can see the red dots. These are the potential. These are sites of the neural stem cells, and you can see that these numbers are the percentage of patients that get brain tumors in these areas. For example, people get about 40% of the GBMs in the frontal lobe. But there is a small but very important region called brainstem, also called pons, and tumors can develop in this region too. Now, unlike this area, that's mostly cerebral cortex, where when tumors develop, in many cases the tumors can be resected, meaning a surgeon can actually go in and debulk, meaning take the bulk of the tumor out. But if a tumor develops in the pons, the brainstem, because this area is really critical that regulates our heartbeat, our respiration, this area is inoperable. So if a tumor develops here, surgery is not an option. So it's basically chemotherapy and radiation. And this, unfortunately, children get this tumor in the brainstem, and because this area is called pons, the tumor is called diffuse, and the tumor is actually very diffuse. It doesn't have a boundary. It's called diffuse intrinsic pontine glioma. As you can see, this is survival of children with pediatric cancer. This 5-year survival rates. You can see that we have been pretty good. For example, in retinoblastoma, and the overall survival is about 83%. And you can see that this is acute leukemia. It's about 90% survival. With DIPG, it's less than 1%. So it's dismal. The situation is really dismal. Fortunately, it's a much rarer disease than many of the tumors, but once you get it, really, there is no cure for it. So we, in our hospital, we try to understand the biology of this tumor. So this is what you can see. This is an autopsy of a two-year-old child, and we try to derive cell lines from these. So we were able to derive this adherent line and also a stem cell line from this piece of tissue that we obtained from autopsy tissue. And we tested the tumorigenicity of the cells, but before that we tested some of the properties of the cell, what kind of markers these cells express. As you can see that these adherent cells express all, at least the important markers of stem cells, which is nestin, SOX2, but not another marker called OLIG2, which is expressed by these sphere cells. Okay, so as I told you before, we derive two different lines. One is adherent lines that grows in serum, and this is serum-independent line. So it is believed that this type of culture actually allows the more stem-like cells to grow. But regardless of whether this is a stem cell or this is a stem cell, the only difference between the two that we found based on the marker expression that we did is the absence of OLIG2 and copious amount of OLIG2 in these cells. So when we try to forcefully differentiate these cells, as you can see that some of these cells that differentiate expresses the differentiation marker, which is GFAP in this case. And some of these cells that are exclusively green in the nucleus, these are the cells that are expressing OLIG2, and they're actually very reluctant to differentiate. So we think that having OLIG2 is a function of aberrant differentiation. So if you don't differentiate the right time in the brain, then these cells will continue to proliferate and maybe give rise to cancer. So we tested the ability of these cells, both these cells, to form tumor in the brain. And we found that the original tumor of the patient, this OLIC2, was expressed at very high levels in the tumor. So we transplanted these cells to the brain, and these cells were not able to form tumors, okay? And remember, these cells don't express OLIC2. When we transplanted these cells, each and every animal formed this huge tumor in the brainstem. So remember, these cells express OLIC2. So we thought that maybe OLIC2 is very important for tumor formation. And within the tumor, we saw that every single cell was expressing OLIC2. So we took the cells, we silenced OLIC2 and asked if the tumors would form or not. And of course, you can see that the control cells, you see very big tumors in the brain. When you silence OLIC2, it actually really suppresses and delays tumor growth, and it gives a tremendous survival advantage to animals. So from this study, we have published this study. Jane was one of the research technician in our lab, and she was really instrumental in carrying out all the studies. We published this in Neuro-Oncology in 2017. I'm going to stop now for the benefit of time, and I want to acknowledge Rishi, my former postdoc, who did most of the study that was published in Nature Cell Biology about the role of AMPK in brain tumor bioenergetics. Nicole is working on a very important project on accidental deletion of genes, which I couldn't tell you today due to the short time. Brunia is a research assistant. She's helping with various projects. Dami is an undergraduate. She's training in my lab. Yen is a new rotating undergrad. Rajiv is a postdoc in my lab, and Vita is a current postdoc, and she's continuing with the MK-mTOR study, and she has other projects also. And Nishka, she's working on a melanoma project, and she's also an undergrad in our lab. And this is my funding. This is National Institute of Health and several other Cancer-Free Kids and several other institutional support that I got from Cincinnati Children's Hospital and the UC Brain Tumor Center. And these are my collaborators. Without their help, I don't think any of our studies would be successfully done. So I'm really indebted to my collaborators, collaborators inside Cincinnati Children's Hospital and throughout the world. And I want to end with this slide. We had a lot of fun. We went to this Mexican restaurant for lunch. This was addition during the summer. It was Jose. He was a rotating graduate student. Priyanka is also a graduate student. He works in our collaborator, but he also gets some stuff done in our lab. And Ben was a high school student, and he came during the summer as a trainee, and he's now an undergrad at Columbia in New York. All right, so thank you very much for attention. And if you have any questions, please feel free to ask me.
Yeah, sure. Can you just, yeah. So it's, can you double click on the screen share, please?
Okay.
Yeah, here we go. Thank you. So it's a good presentation. So I'll have just one question, like it's general, maybe I do not know much about the subject. So you are talking about most of it is AMPK activation of these various transcription factors, right?
Right.
So AMPK is in the cytosol, if I'm not wrong.
Yeah, it's cytosolic is also nuclear.
It is also nuclear. So you see like any difference in those, like when you get the activation of because most of the activation of transcription factors.
Right.
Has to go there, more of it should go there, or whatever is there get phosphorylated and then activates the transcription factor.
Right. It's a great question. Actually, for transcription factors to get phosphorylated and activated, the kinase doesn't always have to be in the nucleus, because transcription factors go in and out of the nucleus. In fact,
in and out of, okay.
In fact, you can actually translocate proteins, including transcription factors, to the nucleus by phosphorylating them in the cytoplasm. Okay, so for example, we found that Krebs, for example, we found that if you actually, in a normal cell, there is some Krebs in the nucleus, some Krebs in the cytoplasm. phosphocrab in cytoplasm. But the moment you cause AMPK activation, we found that a majority of phosphocrab is actually present in cytoplasm. So this actually clearly shows that these transcription factors can shuttle between cytoplasm and nucleus, right?
Those phosphor, because so you are affecting these phosphorylated Krebs affects the transcription, right?
Correct.
Of other genes.
Correct.
Downstream. So do they go to the nucleus then? Like I mean like just I wanted to just get the idea of like, hey, how does it happen, balance?
Absolutely. So phosphocrab goes to the nucleus, and it's Krebs is a very important transcription factor. It's been known for a very long time. So what we found is that the active crab, the phosphocrab, actually binds to the promoter region of GABPA, the transcription factor that regulates mitochondrial function, and it also binds to enhancer region in the HIF1 alpha, and potentially that's how it actually promotes HIF1 alpha.
Okay, that's it. I think you need to rush. I guess it's almost time. So thank you very much, Dr. Dasgupta. I really appreciate it, and look forward to your more work on repeated journals. And good luck.
Okay, thank you. Thank you very much.
Thank you.
Bye.
Bye.